I'm Sadhana, working as Assistant Professor in the Department of Pharmaceutical Engineering, B.V. Raju Institute of Technology, Narsapur. On today's session, I'd like to discuss about the course introduction of Industrial Pharmacy. So here it includes 5 units. So in the first unit, we'll discuss about the pre-formulation studies. In the pre-formulation studies, mainly we'll discuss about all the physical chemical parameters of the drug molecule as well as some pre-formulation aspects which are studied before any formulation if you would like to do. Say for example, if you would like to manufacture a tablet, initially we need to study the physicochemical properties of the paracetamol. We need to study the physicochemical properties of paracetamol as well as we need to study the solubility studies and bulk characterization and stability analysis of the specific paracetamol tablet. Why? Why we need to study the all aspects of the paracetamol? Because the paracetamol was available in different forms. So for example, it is available as a tablet or in solution as well as suspension as well as parenteral route of administration. If we would like to prepare any drug into a dosage form, which route of administration is suitable for the specific drug initially we need to characterize. Then only we can able to prepare any dosage form for any drug. For that, the pre-formulation was necessary. So in this pre-formulation study, first we will go with the solubility analysis. So we need to measure the solubility of specific drug throughout the GI system because the GI system consists of 1.2 pH, 4.5 pH, 6.8 pH and 7.4 pH throughout the GIT. So the 1.2 pH in the stomach, 4.5 pH in the duodenum, 6.8 in small intestine, 7.4 in large intestine. By checking the solubility, initially we will prepare which place is suitable to solubilize and release drug in the GIT. So for example, if the drug is soluble in 1.2 pH, we will prepare a dosage form. That dosage form should dissolve in the stomach and release the medicament in the stomach and absorb to the stomach. If the drug solubility is more in 6.8 phosphate buffer, say for example, then we will prepare a dosage form. After oral administration, immediately it will go to the small intestine. Then there it will solubilize and it will absorb through the small intestine. So that's why initially we need to go with solubility studies. With this solubility study, we will check the effect of pH, pKa, temperature for the solubility. As well as we will go with co-solvency. If the drug is poorly soluble in water, we need to improve the solubility by adding any co-solvents, which we called it as co-solvency. So we need to discuss about the co-solvency and drug dissolution parameters. All the things we will discuss in the solubility studies. So after completion of solubility studies in pre-formulation, we will go for the bulk characterization, which includes all the parameters in micromeritics, which includes the shape, particle size, bulk characterization, bulk density, porosity, everything. So why all this bulk characterization means? While preparing a dosage form, so it need to travel more because from hopper to punching machine, the mixture of drug and other components will come from hopper to the punching cavity to prepare a tablet. So there the flow properties are more important. So that's why including the flow properties, we'll check the angle of repose, bulk density, tapered density, porosity, compressibility index, everything in the preformulation study. Then we'll go to the stability. In the stability, we'll study the solution state stability and solid state stability if the drug is in solution form or solid form. What is the stability of the drug? So, say for example, when we expose it to the external environment, so I'll take a drug and I will openly keep in the lab. What will happen to the specific drug? Everything comes under the stability. And we'll go for drug excipient compatibility. Means if you would like to prepare a dosage form, we need to add many excipients with API. Why? Why all those excipients? So maybe for example, some drugs which are bitter in taste. If I would like to prepare a oral dosage form, I need to add some sweetening agent, which is the excipient. And to attract the patients, we need to add some colors. To improve the patient compliance, we need to add some flavors. So like that, different excipients we'll use to prepare a dosage form. So that's why initially we need to check the drug excipient compatibility. So all this comes under preformulation studies. So in unit 2, we'll discuss about tablets and tablet coating. So in the tablets, we'll discuss different types of tablets, mean to say classification and different techniques. What are the excipients used in the manufacturing of tablets? And what is the methods used for preparation of granules? Mean to say before preparation of tablets, we need to prepare granules to improve the stability and flowability of the drug. What are different granulation techniques? And what are the equipments used for the granulation? And what are different manufacturing process involved in tablet manufacturing? And what is the physics involved in the tablet manufacturing? And tablet manufacturing equipments, what are the specific parts in the tablet manufacturing equipment, which is nothing but a tablet punching machine and evaluation tests for tablets. All these things are discussed in the tablets unit. And in the coating, we'll see different types of coating. Some tablets are in blue color, red color, mainly multivitamin tablets which are available in red color, sweet taste, like that. So we have two types of coating. One is sugar coating and another one is film coating. So to improve the compliance and to improve the stability and to produce an elegant appearance of the tablet, we'll go for printing. So in this tablet coating, we'll discuss about what are the different parameters which are required for a tablet to coat, and what is the process of sugar coating, what are the materials involved in the sugar coating process, and what is the film coating, different types of film formers which are used in the film coating. Everything we'll discuss in the coating. Next, the unit 3 is capsules and microencapsulation. As like tablets, capsules are so a single dosage forms where the medicament is enclosed in a capsule shell. So in the capsules, initially we'll start with preparation of gelatin. So how the gelatin was prepared, the outermost shell of the capsule which is prepared from gelatin. How the gelatin was prepared, what are the sources, and then from the gelatin, how we will manufacture capsule. So here two methods are there. For hard gelatin capsules, initially we need to prepare a shell, then we'll fill the drug. For soft gelatin capsules, directly while preparing only we'll fill the drug. So how we will manufacture a hard gelatin capsule, what is the method, and different filling process in the capsules, everything. Whatever the filling process, whatever the filling equipment which are used for the capsule filling, different types of filling also we'll study in this chapter. And we'll go for evaluation tests for capsules. Different evaluation parameters are there, size of the capsule, moisture content of the capsule, how much amount of the drug, content uniformity, everything is will be covered in the capsules topic. In the microencapsulation, by the means of stability and for increasing the solubility, we will go for microencapsulation techniques. So here we will coat the core drug with any polymer. If I would like to increase the solubility, I need to coat a water soluble polymer, or I need to improve the absorption time. I need to sustain the release. I'll coat a polymer which is water insoluble. Like that, for improve the stability, for improvement of solubility, for improvement of dosage form formulation, we'll use the microencapsulation. In this topic, we'll study what is microencapsulation, what is a core, what is a coat, what are different polymers used for the microencapsulation, as well as what are the techniques, different techniques in the microencapsulation used for preparation of microcapsules. All these things comes under microencapsulation topic. And in unit 4, we'll discuss about the liquid orals. In the liquid orals, we have two types. One is solutions, which is a homogeneous preparation, and another are heterogeneous preparations, which are nothing but emulsions and suspensions. So the basics of the emulsions and suspensions will cover in the physical pharmacy. And what are the manufacturing techniques, industrial manufacturing process, what are the methods involved in the preparation, different types of excipients, what are the equipment, all these things we'll discuss in the liquid orals, as well as we'll discuss about the evaluation test for the liquid orals, whether these are solutions as well as suspensions and emulsions. All these topics will cover in liquid orals. And in the 5th unit, the parenteral products as well as aseptic dosage forms. So in the parenteral products and aseptic dosage forms, initially we'll discuss about the aseptic techniques. Why? What is the aseptic techniques? So the parenterals are the dosage forms which are directly enter into our blood circulation by means of injecting through a needle. So that's why there is no microbial contamination should cause in the parenteral preparations. So that's why initially we need to maintain a aseptic environment to protect the formulation from the microorganisms. That's why initially we'll produce a aseptic environment. Then we'll go for different routes of administration in the parenterals. So we have different routes like intravenous, intramuscular, subcutaneous, intradermal, everything. All the types of parenteral administrations we'll discuss. As well as we discuss about different manufacturing techniques and what are the excipients used in the parenteral formulations and what are the manufacturing techniques for the parenteral formulations, as well as we'll go for evolution of parenterals. Everything we'll discuss in the unit 5. So these are the course objectives. So first objective is to impart a basic knowledge on the pre-formulation studies of the dosage form preparation. So we already discussed about the pre-formulations. So we need to get a basic knowledge on the pre-formulation. Next, second unit to understand different classifications of tablets and tablet techniques, granulation techniques, and what are the compression machinery and what are the coating equipment used for tablet coating, everything. So it comes under the course objective 2. In course objective 3, we'll discuss about, so what is the course objective is to provide the knowledge on the materials and methods preparing different types of capsules and techniques for microencapsulation, everything we will discuss. And that is the third objective in the industrial pharmacy. The fourth objective, which comes on to introduce the concepts of types of additives used in the formulations of syrups, which is nothing but solutions, suspensions and emulsions, which is a liquid orals, as well as some semi-solid dosage forms and how we will pack the dosage forms. All these are in the course objective 4. In the course objective 5, to expose the students with to different aseptic techniques, as well as to discuss different aseptic techniques used in the manufacturing of dosage forms and formulation of parenterals. All these 5 are the course objectives for industrial pharmacy. And the learning outcomes. So after studying these 5 units, so the student can able to analyze different pre-formulation parameters in the dosage form design and classify various types of tablets and different compression techniques of the coating and what is the equipment of coating and evaluation of tablets, and to explain about materials and methods are used in the preparation of capsules and filling of capsules and methods for microencapsulation, and to identify different additives in the formulation of liquid orals and semi-solid dosage forms and packaging of liquid orals and semi-solids. In the unit 5, the outline of formulation details of the parental products and discuss about the aseptic techniques for the manufacturing of a parental dosage form. All these are the learning outcomes in the industrial pharmacy. So here I am expecting students' active participation, timely submission of assignment and regular attendance as well as consistent preparation for the assessment. Thank you.
Vishnu Audio Visual Center, like and share and subscribe and click bell icon.